Good morning, everybody. Let's get chat up. All right, hope you had a lovely weekend. And we're going to just spend a few moments going back over trip operon, because I know that's a real tricky one to get a grip with, I think is probably the best way of putting it. Okay, so this bar is pretty simple, right? So the trip operon is, actually, let's just get this chat up. Chat. Is based on negative autoregulation. And so the product of the trip operon, in the sense of what the proteins produced by the trip operon do, is the production of tryptophan, right? That's the whole purpose from this substrate called chorismate. And this tryptophan, once it gets to levels that are high enough, now this isn't an intermediate level, right? So as soon as tryptophan starts being produced by the products of the trip operon, it doesn't shut down expression, right? Because levels are still pretty low. Don't forget as well that we have the consumption of tryptophan in the production of proteins. Any proteins that contain tryptophan, then making of them will reduce the amount of tryptophan available. But let's say this has been going gangbusters for a while and we've got an absolute ton of tryptophan, then that tryptophan binds to the aporepressor, right? So the tryptophan is the co-repressor. And that aporepressor co-repressor complex is now able to bind DNA, right? Because its shape is such that it recognizes the trip operator, trip O. All right, and then that completely shuts down expression. The real tricky bit is attenuation, and this is all about the leader sequence, which is, it's not very well put, but it's about here, right? This is a better diagram for that, right? This leader sequence here, right? And remember, this is a stretch of mRNA, so this is transcribed, and it begins to be translated. And then the transcription and translation occur in lockstep, right? They occur at the same time. Well, obviously, once enough mRNA has been produced. And this leader region, this mRNA, has two possible. Yeah. I don't know what that was about. Maybe I'm allergic to bacterial genetics. Thank you, Jeremy. Ooh. Thankfully, I wasn't so violent that I nutted the computer. That would be embarrassing. Okay, so the leader sequence, this 5', well, it's not untranslated because it is partially translated. But this leader region has two possible conformations, right? So we talked about the formation of RNA secondary structures before. This is a really good example of that. Sorry, let me just mute. Yeah, this is like day in the life of a professor or something. This is anyway. There are two possible conformations. We can have region 1 and 2 bound together to form a hairpin loop. We can have region 3, and if we do, then region 3 and 4 also are able to form a hairpin loop. The second possibility, sorry, itchy nose, is that if region 1 is not available, region 2 and 3 form a hairpin loop before 3 and 4 can, right? So remember, these hairpin loops form as soon as the bits that stick together are transcribed, are produced in mRNA. So if there's nothing stopping 1 and 2 from forming, they will form as soon as they're produced. And so if 1 and 2 are formed, that means that 2 and 3 cannot form. Right, so they're essentially these two different conformations are mutually exclusive. You can only have one or the other. You can only have 1 and 2 bound or 2 and 3. Okay, so it's kind of like essentially telling you the same thing I told you on Friday, but telling you in a different way. And so the key part of the attenuation of the trip operon is which of these conformations are formed and how is the choice controlled. And so sorry, got a cat alarm going off. If this conformation forms, 3 and 4 are a terminator loop, right? So we have this series of U's, which pauses RNA polymerase, remember from intrinsic termination before. And if the RNA polymerase is paused over these U's, then the formation of this loop kicks it off of the DNA and the RNA, right? Causes a dissociation of RNA polymerase. So this is the terminator loop, right? Or attenuator sequence. It's another way of calling it. Does that make sense? Good. And this will form if 1 is available to pair with 2. That's the key, right? Now, if 1 is not available to pair with 2, and you know why that is, but we'll go back over in a little bit. It's down to these trip operons here. If 1 is not available for pairing, 2 is just kind of flapping around in the breeze, right? It's just kind of waiting around like, oh, who am I going to hang around with? Until 3 is transcribed, and obviously 3 is transcribed before 4. So 2 and 3 will form a loop, and that prevents 3 and 4 from pairing up. So this no terminator loop is formed. RNA polymerase is not punted off of the DNA. And so the thing that basically changes this from one to the other, right, one possibility to the other, is whether or not a ribosome is paused on region 1. That's it. And that ribosome will pause if there is insufficient tryptophan available for charged tRNAs with tryptophan to come in to allow the synthesis of that short polypeptide sequence. So the ribosome acts as a well, actually, in a way, these two trip codons are the sensor, right? If there is insufficient trip available because it's not being produced enough or it's being consumed too fast, then the ribosome will not be able to translate this short sequence. If it can't translate this short sequence, it hangs out over these trip codons and prevents 1 from base pairing with 2 in this conformation. And so 2 is free to base pair with 3, 3 and 3, which again prevents the formation of this terminator loop. So if we look at it in a bit of a different way, over here, right, this is looking at it in a series of events, right? So you can see RNA polymerase is happily puncturing along the leader sequence, transcribes region 1, and as soon as as it progresses along, now region 1 is available for translation. Cats. And that ribosome, those two move along at the same rate, more or less, right? And so region 2 isn't transcribed yet as the ribosome starts translating region 1. Okay? So now the RNA polymerase has moved further along, It's transcribed region 1 and 2, and it's now in region 3. Okay. However, the sorry, getting distracted by cats. Amor, you're going to have to go and see to those cats. Very distracting. All right, so this ribosome is now paused over these 2 trip codons. You can see the little polypeptide sequence coming out. This polypeptide doesn't do anything. It is purely there as a byproduct of this sensing mechanism. Right, and so the polymerase pauses. You can actually see this kind of dragged along because this is now the RNA essentially. As RNA polymerase transcribes region 3. So now 2 and 3 are available to pair because they have complementary sequence, and the 2-3 loop forms. And this is before RNA polymerase transcribes region 4. I'm really going crazy with the rhyming here. 3 and 3 and before and 4. Huh. I should be a poet. Anyway, so if that happens, then the terminator loop doesn't form. RNA polymerase finishes this little leader sequence and starts transcribing. Come on. TRPE, which is the first structural gene. And so this whole polycistronic mRNA is produced, translated by ribosomes, turned into the enzymes that generate tryptophan. Right, now tryptophan levels start increasing because we are having transcription and translation of the trip operon. And now we get to this situation, right? So we have the same thing again, RNA polymerase, ribosome binds to region 1, starts translating it. RNA polymerase is transcribing region 2. Now, as the ribosome gets these 2 trip codons, well, you know, there's actually plenty of trip tRNA around. So the ribosome proceeds onto region 2, right? And as a result, region 2 is not available to base pair with region 3 once region 3 is produced, right? So now region 3 is going to be hanging around, right? Waiting for something to bind to, and then region 4 is produced, and as a result, it kicks the RNA polymerase off of the DNA. And that terminates, or rather attenuates, transcription so that the trip operon is not transcribed. Now there's going to be a kind of, it's almost like a probability basis here, right, of whether or not that RNA polymerase continues on into the trip operon. And that's basically down to the probability of there being a charged tRNA trip around for that or 2 actually, for that ribosome to continue onto region 2. And so even when trip operons are increasing, there's a probability that at that point in time, there is no charged trip tRNA. And so translation pauses, termination doesn't occur, and transcription continues. But as those levels of tryptophan get up, the probability that there's going to not be a charged trip tRNA around gets lower and lower and lower, and so attenuation becomes more and more and more common, and overall transcription of the trip operon decreases. So it's one of those things which can appear very, very complicated from, you know, the overall, but if you think about the individual steps of what's going on, it's actually not too bad, right? And so that's really all that we're going to do for bacterial control of gene expression. Does anybody have questions? Questions from last week or questions from today? hopefully going over this for a second time after a period of time and in a slightly different way will have helped your understanding of what's going on. Sorry, just got to blow my nose again. Feel free to type up any questions you have while I'm doing so. Cool, well that's good to hear, Jelani. But don't forget that it's sometimes it appears easy to understand as I'm explaining it. But the real trick is making sure that when I ask you questions about it in the future, in like 2 weeks' time or a bit less, you still understand it then, right? So that's the tricky bit, right? So make sure that you can essentially, without reference to your notes, be able to draw out this process, be able to answer questions from your peers, right? Study groups are really good, even in this weird age of online stuff. Actually, it might even be easier to form study groups online than it will in person. And also try and find questions that you can quiz yourself with about this stuff. Because again, right here and now, I'm sure most, if not all of you would be able to answer questions about this, right? Because it's right front and center in your mind. You've just had it explained, for a second time, but that will fade a little bit over time as that kind of immediacy of that memory dissipates, right? You have to build on that and keep kind of adding, kind of essentially it's like building a house, right? You throw up the sticks first, and then you keep adding to that until you get a robust structure at the end. Okay. Oh, what's going on over there? Cool beans. All right. Woohoo. Didn't mean to jump straight in there. Okay, so the next thing is actually, in many ways, a much, much larger and more complicated topic, and that is of eukaryotic gene regulation. And so part of the reason why eukaryotic gene regulation is more complex is because it occurs at many, many, many more levels, right? So prokaryotic control of gene expression is largely at the level of transcription, by and large. So there's something ticking in the kitchen, and I'm trying to figure out what it is, half-heartedly. And also, each of those levels has kind of knock-on ramifications, right? So you can control gene expression at or the production of a functional protein. You can control it at real, like, zoomed out level, right? In terms of, okay, I'm going to shut down whole swathes of the genome, right? That region of chromosome 2, it's off, all of it, right? So any genes that are in there will also be off. Then you can go down to the individual gene and control it at the level of transcription, of which there are many, many more bells and whistles involved, right? So now we have, and this is just a, God, just give me a second. This is driving me nuts. Sorry about that. Now I know what's happening. My wife's making some iced tea. Ha! It's a long, long time since I remember that. Anyway, all right. And you can see here there are basically many, many more regulatory elements in a promoter in a eukaryotic gene. We have Enhancer sites, right? So these increase transcription. We have our basal regulator, right, which contains the Tata box and is all about, RNA polymerase binding. We have repressors, which compete with enhancers and may even occlude some of the RNA polymerase binding sites. There's a lot going on, even just at the level of transcription. And then you have everything after that, right? So if you remember back to that kind of linear path that I drew a while ago, must have been before we got into bacterial gene expression. We have, actually, here you go. Here's a bit of a recap or quizzing for you. What different things, and you don't have to say them in order, can you think of what other ways in which the formation of a functional protein is controlled? How much of it you have, let's say, or whether you have it or not in a particular cell. What other things, other than the control of transcription, control how much functional protein you get from a gene? Here you go. Spit them out. Tell me, what can you remember? What other steps? Think about all of the steps of turning the information in DNA into a functional protein. What other processes happen along the way which can affect that? Come on, this is a good way of, this is a kind of in a sense, a favor from me to you to help you revise and study this subject. Okay, what other things happen between DNA and functional protein? Modifications? Good. Of what? What are we modifying? I can think of a couple of things. And the Golgi apparatus? Yes, right? So by the time something gets to the Golgi apparatus, what macromolecule are we talking about? Out of our three possible, DNA, RNA, protein. Which one goes through the Golgi apparatus? You're on the right track for sure. No. mRNA is going to be in the cytoplasm or in the rough endoplasmic reticulum or thereabouts. Not actually in it, but near to it. So if it's not mRNA, it's not DNA, it is, yes, protein. All right, so protein modifications, of which cleavage and adding things and taking things off and stuff like that is a big one. Okay, so that's kind of right at the end of that process. What else? That's a good one. Thank you, Jeremy. What else? Oh, yeah, just don't worry, I make spelling mistakes all the time. Okay, so we've got protein modifications. What else is going on in that series of events from DNA to functional protein? How do you get protein? Functional or not? What's the process by which you get that? Come on, it's all part of the central dogma sequence hypothesis. How do you turn DNA? Ooh, almost told you. Other than transcription, sure. So, okay, in eukaryotes, what do you get at the end of transcription? Pre-mRNA. Okay, so how do you get from pre-mRNA to functional protein? What steps must occur? Well, the key is pre-mRNA. So what's the process that turns pre-mRNA into mRNA? There's a series, 3 different events. I'll give you a clue. There's capping, 5' capping. As soon as the cap appears, once the pre-mRNA has been cleaved, there's poly A, tail addition. What's in between? What's the other thing? It's one of those things that you do with old school film reels and things like that, where you cut out different bits and stick things back together again. That's part of it, but what's the general process called overall? You cut the 5' end, loop it. You cut the 3' end. What are you cutting out? What bit of mRNA are we cutting out? Or pre-mRNA, rather? Introns, right. So the removal of introns is called what? Splicing. That's it. Thank you, Sophia. Exactly. So we've got splicing, right? Is part of the control of gene expression. Okay, now we have an mRNA. What are we going to do with that mRNA? What happens next? Now we have a functional mRNA that has been exported from the nucleus. What happens to it now? What is it used for? What's the next step in gene expression once you have a functional mRNA? Also begins with T. Yeah, to produce a sequence of amino acids. What's that whole process called? It's what you do if you turn what's in one language into another language, say Spanish to English or Suomi to, to Cyrillic or anything, Japanese to South African. Translation, that's right. Okay, so if that mRNA is degraded before it can be translated, will you get a protein produced from it? If that mRNA is degraded by the cellular machinery? Yes or no? 50% chance of getting one of them right. No, that's right. Good. Correct. I'm assuming that was not a guess. But no, you don't get protein produced, right? So the amount of protein produced from an mRNA depends on how long that mRNA is around for and available for translation. Okay, so that's RNA stability is another big part of this. Okay, so now what about translation? Is translation automatic in eukaryotes? Does it just happen if an mRNA is present? Or is there other stuff around which has to promote translation? think about the process of translation. The promoters in the gene, that's for the control of transcription. And the control of translation, think back to how the small subunit assembles on the mRNA. We need cap binding proteins, and we need poly A binding proteins to help that process, right? So if you interfere with any of that, the ribosome is not going to assemble, right? And so you can actually control the amount of translation by controlling the initiation of translation as well. Okay, so control of translation it's not as well, actually, I don't know whether I don't think it's quite as complex as a control of transcription. There's a few less bells and whistles that you can play around with, but there certainly is quite a degree of control of translation. Okay, and then the last one, what happens if that protein gets degraded before it's used? Do you have any functional protein? No. Okay, so think about this as a sum, like an equation, right? Is DNA open or closed? Is it available for transcription? Do you have sufficient enhancers and insufficient repressors available for transcription to start? How are you splicing your mRNA? Is your mRNA stable? Is it going to be translated? Is your protein going to be degraded? Is your protein needing to be modified? Essentially, you have like 7 different things at least where you have to essentially add up the outputs of all of those to get the answer of how much functional protein you have. So now you can see that this is far, far more complex than control of prokaryotic gene regulation. However cool and interesting it is, and it is without a doubt, in many ways it's much more elegant than eukaryotic gene regulation. But there's a lot more going on in eukaryotes because of all those different levels. Each one has its own level of complexity, right? And different outcomes. And essentially you have to sum all of those up in some kind of funky complex equation to be able to figure out exactly how much protein you're going to get out of it. Right, and so this is summed up really nicely in this diagram, which basically kind of takes all of those different steps and puts them in order, right? And first, what we're going to be dealing with is chromatin modifications. Oh, I'm sorry. Needing some coffee, right? And so this is going to be like the first step, because obviously without this, in the right format, you're not going to get transcription at all. And typically we think about this in terms of a signal from outside the cell, right? So a cell is told, you're going to do such and such, or you're going to be such and such, right? And that signal has to make its way from outside the cell, through the cell membrane, and then into the nucleus somehow, through either a chain of signaling events or something that goes there directly. Something like retinoic acid is a good example of a direct thing that's actually found a lot in acne medicine. I don't know if any of you have ever had, shoot, what's it called? Can't remember off the top of my head what it's called, but there's some real serious acne medication that if you're a woman, you have to basically go on birth control while you're on it, simply because it is so potent that if you do conceive, it can really mess up the development of your child if you are taking it at the same time. So anyway, that signal has to get in somehow and prompt a response. That response typically being gene expression, right? Production of a functional protein. And these are just the steps involved in that, right? Oh, and also forget transport to where it's actually going to be. That can also be a important step. And at the end of all of these, you have a protein that does something, right? Whatever that might be, being part of the structural support of the cell, could be in a metabolic pathway. It could be a protein that moves things from one place to another, right? From, the cell body out along axons or dendrites or whatever, if it's a neuron. Could be a signaling molecule that's involved in the generation of that signal, which tells in turn other cells what they should and shouldn't do, right? And so this is a long and complex kind of set of conditions, essentially. And that's why we're going to take a lot of time to go through these, at least most likely this week, to be honest. We'll see how we get on. And so essentially, I should just move this up here, really. So these are two cool, I love microscopy. I really do. Let's bring that even bigger. So this is a picture of a neuron up here, right? So this is a neuron in vitro, right? So this is growing in some kind of substrate. And this is what's called a confocal micrograph. So this is a picture that's taken with a laser. And that laser is focused on kind of different layers of the cell, right? If you think of a cell as a three-dimensional object, essentially a laser is scanning each level in turn, and each level has a picture taken of it in turn, and then they're stuck together in this super beautiful picture. And I don't actually do much confocal microscopy. We don't have one here because they're really, really, really expensive. The one I was trying to get a grant to buy would have been about $500,000. Yeah, just for the microscope. And the maintenance plan was $10,000 a year. So that tells you how they're very, very crazy. Anyway, so I don't know whether this is by immunostaining or fluorescent proteins, because I don't know exactly where this picture came from. But essentially what you've done here is you've stained microtubules, right? So these are the structural parts of the cell that gets products from the soma, which is kind of the cell body essentially, out to the tips. And then you have these growth cones, which are basically how neurites, axons, and dendrites grow to their destination, right? They basically kind of creep along like that through cells and around them and along a surface until they get and basically responding to external signals, right? This is one of my fun things to talk about in developmental biology. But they grow in a particular direction based on the sum of the signals that they're getting, right? And that's through the formation of these actin cytoskeletons, right? And those collapse essentially where they're not getting a signal, and they get stronger where they are, right? And so this is a neuron. And you can see the nucleus is probably about here, would be my guess. And this is a keratinocyte, right? So this is basically a cell which forms very rigid structures in your hair, right? Or your fingernails, other places like that which have keratin, right? And that's formed by these cells. They almost look like plant cells, right? They're very rigid, right? There's a nucleus. They're very planar, right? They're flat, and they're stuck together very, very tightly, right? They form a physical barrier essentially. And this is essentially all the fancy juju that you have in shampoos and stuff and facial creams and things is supposed to be about nurturing and helping these cells. But this cell and this cell have exactly the same DNA. They have the same set of instructions to produce one or other of these. So how are they different? Why are they different? Right? And it really comes down to, well, actually a lot of things. But the end of it is, which of those genes do they express? There is a certain panel of genes in keratinocytes which make them keratinocytes. And subsequent genes that are expressed for them to be able to remain being keratinocytes, to be able to, carry out their function, including proteins that make them stick together very tightly at their edges. And that panel of genes that are expressed is very different from the panel of genes that are expressed in neurons, as you can imagine, right? There will be genes which are expressed in both of these, right? Things involved in DNA replication and repair, in transcription and translation, right? These cells will both have cytoskeleton and actin in them in different ways and different places, but they will both be expressed in those genes, right? So there are genes which are shared between them, but there are also genes which are very much differentially regulated. And that differential regulation is the interesting bit, right? How do these become these and not these? And if you think about it from the point of view of medical biology, right? If you want to make these, let's say if you're researching how to fix people that have had traumatic spine injuries or brain injuries, right? How can you take some of their stem cells from that patient and grow a bunch of neurons and then implant those neurons back into that individual so that you can repair that break in their spinal column, for example, right? So that being able to actually create these cells is a big driver of understanding how they are created, right? So that we can do that externally and then, put those cells back in. It's like baking, basically. Okay. And actually, there's been a really cool, like many over the last few years, developments on generating balls of cells which produce cells on their own without external kind of cues. Because a lot of the reason why these cells are what they are is their context. What cells are around them? Glia, for example, in the case of neurons. Not quite sure in case of keratinocytes, right? So anyway, formation of organoids is also super cool. Okay, so anyway, chromatin is a big part of that, right? So a lot of the genes which are involved in neuron formation are grouped together, roughly, not in the same way as operons are. Oh, sorry, not the same way as operons in bacteria, much more loose associations than that. But they'll typically be on at least shared regions of a chromosome. And in keratinocytes, those regions of the chromosome are shut down. And what we mean by shut down is the formation of heterochromatin, right? And heterochromatin is, let's move that down. That's funny. Let's move that down there. Is the formation of compact chromatin. And what we mean by compact chromatin is chromatin or DNA that is tightly bound to proteins, histones, and not available for transcription. And that's kind of based on a lot of different levels. One is how tightly DNA is bound around those proteins. And two, how tightly this whole complex is bound around each other, right? So it's not just this open beads on the string, it's a very tightly compact kind of knot or rope of chromatin, right? And so that's thought to be the default state of chromatin, right? Chromatin is naturally squished up and then becomes told to open up, right? And the net result is that if chromatin is compacted, the regions that are required for gene expression, for transcription, are less available, right? So those proteins, right, the Tata box binding protein, enhancers, things like that, are unable to get to, oh, here's the TVP binding site, are unable to get to access that DNA. So in the last few minutes, I want to talk about some words, right? Some language that we're going to use with this. And so when we talk about chromatin, that chromatin isn't this way just by chance, right? There are tags or bits of information that are added to that DNA and protein, which alter its state, right? And these are called epigenetic modifications. So epigenetics is essentially modifications that change gene expression without changing the nucleotide sequence, right? So actually, this is a really kind of neat thing to think about, is that you also inherit these from your parents. So part of what makes you you isn't actually your DNA sequence. It is the epigenetic modifications that you inherit from your parents and the ones that you have programmed into you by your mother. This is actually a real cool thing. Just for uh, the ladies present, which actually I think almost all of you are. Oh no, JM's present. Here you go. So JM, you can just not pay attention now if you wish. But and actually, I mean, presumably JM, you have a mother, you might have a sister, and sometime in the future you might have daughters. But for the ladies present, this is actually really, really crazy. You were an embryo, not an embryo, an oocyte. What am I talking about? You're an oocyte, right? So an egg inside your mother, while your mother was inside your grandmother. Because as, yeah, I know, isn't that crazy? So women make their oocytes during embryonic development, as well, more as a fetus, right? While you're inside your mother. So Sophia and Jeleny, you contain oocytes in you that you made while you're inside your mother. And so those oocytes, right, contain DNA, obviously, right? The maternal DNA that you'll contribute to the next generation. And that maternal DNA can be modified with epigenetic modifications. So essentially, part of the way why you are the way you are is based on the experiences or the conditions that your grandmother experienced while she was pregnant with your mother. So, yeah, I know. Next time you see your grandmother, if she's still around, hopefully, you can ask her, "Hey, Gran, what was life like when you were pregnant with my mum?" And she might say, "Oh, it was absolutely lovely." And it's like, "Oh, that's why the way I am, the way I am." So yeah, it's essentially 3 generations are present in one individual at a given point in time. Mostly it's I think it's the second to third trimester. I think probably more the third trimester. But yeah, so if your mother or if your grandmother, sorry, was experiencing severe stressful conditions, be it famine or civil war or something, right? Then those conditions will have imprinted on you through your mother as she was vitro, in vivo, sorry, in utero, in your grandmother. And those are epigenetic modifications. So the DNA sequence doesn't change, but how accessible that DNA is to transcription does change. Anyway, I've gone off on too much of a tangent. I just thought that was really cool. I love talking about that stuff. So we're out of time. We'll continue for, yeah, so in that sense, yes, that only affects females because males produce their gametes once they reach puberty. Yeah, so that's the key point is that the gametes that you're going to use to create the next generation are in females present in you when you're a developing fetus inside your mother. For males, that isn't the case because those gametes are only produced on reaching puberty, basically, and the full development of the testicles. And also, there's a constant turnover, right, of gametes in males, whereas in females, um, all of the oocytes you will ever have, you've already have them by the time you're born. Yes, the H1 histone is considered part of the core histone molecule. That probably going back to the question in the exam where there's 9 proteins and 145, 147 base pairs. Yeah. So it's the 8 histones that the DNA is wrapped around, and then the H1 histone kind of holds them in place in part. Cool. Yeah, I know. That wasn't meant to be tricky, although it kind of came out a little bit like that. Is actually you got to think back and what is everything involved, and it's the 4 pairs and the H1 histone. Cool. All right, everybody, we'll continue this on Wednesday. So see you all until then. Stay safe and study hard, and I'll see you on Wednesday.